Assalamu alaikum dear students. Okay, so today's our topic is photorespiration. Now, as I have already completed this photosynthesis, like this light reaction and dark reaction. तो जो हमारा आज का नया टॉपिक है फोटो रेस्पिरेशन। तो सबसे पहले हम समझेंगे यहाँ पे कि फोटो और रेस्पिरेशन means photo means light and respiration means you know that what is respiration. So it means respiration in presence of light. Normally what you see the normal respiration, light has no role in respiration, right? But here this respiration is a special type of respiration and where light in the presence of light, and this type of respiration it takes place in green parts of the leaves, okay, or green parts of the plant, or you can say especially in leaves. But it takes place, okay, so this term was given by Krotko and Decker, and it is a special type of respiration and that part takes part in green parts of the C3 plants in the presence of light. Okay, so remember that photorespiration takes place only in C3 plants. Now what are C3 plants? C3 plants क्या होते हैं जो जिनमें C3 साइकिल हो जाता है। मतलब अभी तक हमने कल जो पढ़ा था डार्क रिएक्शन, फोटोसिंथेसिस का जो सेकंड फेज है डार्क रिएक्शन, उसमें हमने पढ़ा था कि this dark reaction it is also called as Kelvin cycle because Kelvin Benson cycle, and it was discovered by Kelvin. So this Kelvin cycle is also known as C3 cycle because the first compound that was found here is that it is 3-phosphoglyceric acid, which is a 3-carbon compound. So that is why it is known as C3 cycle. Second point is that this C3 cycle is present in some plants which are known as those C3 plants. So these C3 plants they possess this photorespiration. Not all the plants but only C3 plants. This photorespiration present in C3 plants. And then this C3 this photorespiration ये 3 ऑर्गेनिज में हो जाती है. It takes place in 3 cell organelles. कौन कौन सी? Chloroplast, peroxisome, mitochondria. These chloroplast, peroxisome, and mitochondria they are associated with another. They are closely attached to one another. What happens here actually? It takes place in high oxygen concentration. When the oxygen concentration is high and there is low concentration of carbon dioxide, there is one enzyme which is known as rubisco. So I already told you the function of rubisco. The enzyme rubisco is used in C3 cycle. Rubisco means ribulose 1,5-bisphosphate, carboxylase and oxygenase. So this rubisco normally acts as its substrate is usually carbon dioxide. But when there is low carbon dioxide concentration and higher oxygen concentration, this rubisco enzyme it acts as oxygenase. It shows its oxygenase activity. That means now it will not accept carbon dioxide, but it will accept oxygen as its substrate. So that is why it shows oxygenase activity. Now when oxygen is substituted for carbon dioxide, the toxic product that is known as phosphoglycolate it results. This phosphoglycolate is a 2-carbon compound. So this photorespiration is also known as C2 cycle because phosphoglycolate, when we see later on, it is a 2-carbon compound. Okay, like 3-phosphoglyceric acid, it is a 3-carbon compound. So this cycle was known as C3 cycle. Same thing happens here. This phosphoglycolate it is a 2-carbon compound. That is why this photorespiration is also sometimes known as C2 cycle. Now these plants they metabolize this phosphoglycolate. It is a toxic compound, and plants must use up or metabolize this phosphoglycolate very soon, very quickly. So they metabolize this phosphoglycolate, but for the metabolism of this phosphoglycolate, it extra energy is required. Okay, extra energy is required, and there is also loss of some carbon dioxide molecules. So what happens actually? Carbon is lost in the form of carbon dioxide. So when carbon is lost, it is a fixed carbon. So that means it is a very harmful process. Photorespiration it is a very stressful or harmful process for plants. Now you can see here this is a diagram of leaves. So when there is high temperature outside, there is higher temperature outside, so this stomata which is usually open for this gaseous exchange, okay, that means intake of carbon dioxide and removal of oxygen, and also this transpiration. When there is heat outside, there is a stressful condition, the leaves they close their stomata, right? When stomata gets closed, oxygen remains inside the leaves, it cannot escape, and carbon dioxide cannot enter in. So there is increasing oxygen concentration. Now when there is higher oxygen concentration, this rubisco will act as oxygenase. Okay, so it will use up this oxygen to form phosphoglycolate. These are the three organelles: chloroplast, peroxisome, and mitochondria. It is a real picture, SEM, scanning electron micrograph of this. This is the picture. So here what you see: chloroplast, peroxisome, and mitochondria. These are closely associated, as I already told you. And this photorespiration takes place in these three organelles. This is the actual cycle of photophosphoryl of this photorespiration. So in chloroplast, what happens? First point is that this RUBP, so it combines with oxygen with the help of enzyme rubisco. So ribulose 1,5 bisphosphate, it combines with oxygen to form phosphoglycolate. Now this phosphoglycolate, which is formed in the chloroplast, it is a toxic compound and it must be metabolized. So what happens? Phosphoglycolate, the phosphorus group is removed, dephosphorylation takes place. Okay, and glycolate is formed. Now this glycolate is transported to peroxisome. In peroxisome, this glycolate, oxygen combines with glycolate to form hydrogen peroxide and glyoxylate. Now this hydrogen peroxide also, it is a toxic compound and it has to be metabolized, it has to be degraded. Now hydrogen peroxide is degraded into oxygen and water later on with the help of enzyme peroxidase. Now this glyoxylate, it is transported to mitochondria. It is first converted to glycine, that means an amino acid. Now this glycine is transported to mitochondria. Now in mitochondria, this glycine is decarboxylated and its amine group is removed to form serine. So this is known as the enzyme uses glycine decarboxylase. So this serine which is formed, now another amino acid which is formed in mitochondria, it is transported back to it is transported back to peroxisome, where the serine is converted to hydroxypyruvate. Now this hydroxypyruvate is converted back into glycerate, and then glycerate is back converted to 3-phosphoglycerate. 3-phosphoglycerate used in Calvin cycle, and then through this 3-phosphoglycerate, RUBP, ribulose 1,5 bisphosphate, is again regenerated, which again combines oxygen to complete this cycle. So this is how this photorespiration takes place. Now this photorespiration, does it have this significance? The cost is, what are the costs of photorespiration? In normal air at 25 degrees Celsius, photorespiration decreases efficiency of carbon dioxide assimilation by 40%. That means photosynthesis, the production of glucose, the production of sugars, it is reduced by 40% because of this photorespiration. So photorespiration is a wasteful process. Now this decrease in efficiency results from the use of ATP to phosphorylate glycerate and the other costs associated to this processing. So is there any benefit of photorespiration? Yes, somehow proposed that this photorespiration allows plant leaves to use up excess light energy and reduce photooxidant damage when the plant is water stressed and the stomata are closed. Okay, so this is all about photorespiration. Tomorrow we are going to talk about our next topic, that is C4 cycle. Thank you.